Hello, my name is Gideon Akuamoah Wiafe, and you are welcome to my presentation. I am an MSc candidate at the University of Cape Town, currently based in the Neuroscience Institute. Today, I'm excited to present our study which leverages long-read whole genome sequencing and pangenomics to explore a rare neuropathy linked to variants in the MPV17 gene. So for a little background, the MPV17 is a gene that is associated with mitochondrial DNA depletion syndrome, which is a severe condition typically inherited in an autosomal recessive pattern. Interestingly, emerging reports have linked biallelic MPV17 variants to milder late-onset axonal neuropathies, but confirming pathogenicity in such cases is often challenging. In our cohort of South African patients with inherited neuropathies, 4 probands were found to carry 2 MPV17 variants, that is the PH125TRP and PGLEN36TER. However, prior attempts using exome sequencing, short-read whole genome sequencing, and even targeted long-read sequencing could not establish whether these variants were in trans, which is a key characteristic for recessive diseases. We therefore hypothesize that long-read whole genome sequencing and pan-genome-based approaches could help resolve this ambiguity, particularly in African context. So for our methodology, as I said earlier, 4 probands underwent the various sequencing technologies that I mentioned, but unfortunately, the pathogenicity of the variants that they harbored were not confirmed. So in figure 3, we will see how these sequencing technologies performed upon viewing the aligned BAM files in IGV. But however, due to cost, one sample underwent high-fidelity long-read whole genome sequencing, leveraging the review system by PacBio at Nkaba Biotech here in South Africa. And the reads from this review system are on average 30x coverage. So as we received our hi-fi reads, we developed and optimized an automated Widow-based workflow to analyze our data. And the Widow-based workflow is shown in figure 2. And this workflow processes single samples, hi-fi samples from read alignments to generating statistics on the alignments to variant calling, that is calling short variants, structural variants, copy numbers, and tandem repeats, performing filtering, annotation, and further performing various tertiary analysis. This pipeline required on average 15 hours per sample, utilizing approximately 80 gigabytes of memories, and was deployed on the Elifo Research Computing Cluster. Briefly, we aligned 30x Hi-Fi WGS, that's whole genome sequencing reads, to the linear GRC H38 reference genome and use variant calling tools optimized for long read data to assess the phase of the MPV17 variants. To complement this, we examined the MPV17 locus within the HPRC, that is the Human Pangenome Reference Consortium graph, which includes 94 haplotype resolved assemblies, nearly half of which were of African ancestry. And this is shown in figure 4. So this allowed us to assess the copy number diversity across a broader genomic context. So from figure 3, which is the images or the screenshot from IGV, we can see that our long-read data successfully confirmed the 2 MPV17 variants in question, that is the PH125TRP and the PGLEN36TER. It's actually showed that these variants were in trans, which supports a compound heterozygous state that is consistent with autosomal recessive inheritance relative to the short-read whole genome sequencing and the targeted long-read sequencing, which are above the the long-read whole genome sequencing image in figure 3. So as seen in figure 4, we also confirmed that the MPV17 gene was present in a single copy across all the 94 haplotypes in the HPLC pangenome graph. This finding eliminates concerns about undetected duplications or structural complexities that might confound the interpretation of the 2 variants we discovered in the MPV17 gene. So these results provide robust genetic evidence supporting a potentially pathogenic role of these variants in the observed neuropathy phenotype, particularly relevant for African patients who may present with a typical, well, with a milder form of known genetic diseases. So for our study, we believe that this study demonstrates the power of long-read sequencing and also pangenome analysis to resolve complex diagnostic questions, especially in settings where trial sequencing is not feasible due to cost and resources. It also highlights how African inclusive pangenomic references can enhance our ability to interpret rare diseases accurately and confidently in African populations. Beyond this particular case study, our workflow sets a foundation for broader rare disease discovery in Africa, potentially accelerating diagnosis and enabling personalized interventions for patients who would otherwise remain undiagnosed. So, for conclusions and future directions, we believe that in summary, our findings illustrate the critical value of high-resolution genomic technologies and diverse reference frameworks in rare disease research. Also, our workflow, which is designed for hi-fi long-read sequencing, can be adapted in other case scenarios as well. Thank you to all my supervisors, my colleagues, funders, patients, and families.